abemaciclib, two very large randomized trials of MONARCH 2 with fulvestrant and MONARCH 3 with the aromatase inhibitors, both showed significant benefit from abemaciclib with endocrine therapy. There was a deep dive done into the patient phenotypic subsets, patients with bone-only disease, patients with very long natural histories, patients with very short natural histories, liver metastasis, grade of the cancer. And basically, it was interesting, what was found is that the abemaciclib's greatest benefit in terms of patient subsets were those with more aggressive biology. It worked in all of the subsets of patients, as all the CDK4/6 inhibitors have, but there seemed to be a differential benefit in those with liver metastasis, for example, those with shorter treatment-free intervals who stopped endocrine therapy or who recurred on endocrine therapy, in the adjuvant setting and are recurring, manifesting endocrine therapy resistance, really benefited. And, it just may be that these cancers are more highly proliferative and may have multiple mechanisms of endocrine therapy resistance. Abemaciclib, of course, is a very potent CDK4 inhibitor. It's the most potent of the three CDK4/6 inhibitors, particularly against CDK4. And so in these full throttle cancers, you need continuous target inhibition, potentially. That really has to be put to a randomized trial, but it's a hypothesis. And also you need very strong inhibition of the target, with a potent inhibitor. Abemaciclib also inhibits more than just CDK4/6. It does have some inhibition of CDK2, potentially even CDK9, so some of the other CDKs that can cause resistance to CDK4/6 inhibition. So we know that some of these cancers, for example, have higher levels of cyclin E. Some recent data from Nick Turner looking at Paloma-3, looking at the tumor, the primary breast cancers in patients who ultimately developed metastatic disease and were randomized to fulvestrant plus minus palbociclib. Patients whose primary breast cancers had higher levels of cyclin E, which complexes with CDK2, which is not inhibited by CDK4/6 inhibitors, those patients didn't do as well with palbociclib, as those who had lower levels of cyclin E, where the cancer was more driven by CDK4/6. Those patients did extremely well. We don't know those data, for abemaciclib, but we do know it has some inhibition of CDK2, which complexes with, so a broader mechanism of action potentially as well may help these more aggressive cancers that recur more quickly, probably because they have multiple mechanisms of resistance against endocrine therapy.